All right, everyone. So what we're going to be looking at today in this lecture video is probability and statistics surrounding Punnett squares. So in the last video, we ended by talking about the process of cells segregating their genes into the final product cells, so the egg or the sperm, and then coming together to produce offspring that have different physical expressions or phenotypes. And here what you can see is that when you're putting these gametes together, there's a chance that this gamete would go with this gamete, right, versus this gamete going with this gamete. Okay, so because there are different variations of what could potentially happen, there's a probability of surrounding what actual opportunities these cells are going to have based on the phenotypes that they have. So one thing that Mendel observed in his observations is that from the F1 to F2 generations, so remember filial generations, the F1 is the one that's after the two true breeding parents come together, and then the F2 is when he took those hybrids from the two first parents and put them together. He noticed that in each generation, when he took the F1 and F2, he had about the same number of phenotypic distribution, the same number of phenotypes showing up. So he would see like 3 tall for every 1 short that was there. And what he ended up realizing, because Mendel was a mathematician, is that it was following basic rules of probability. So remember that probability is the likelihood that an event will occur. Okay, so whether or not, say, you're going to get A on a test, or the probability of winning the lottery. Okay, all of these things have a various likelihood depending upon a few different things, like studying or buying the right ticket. But using these principles, we can predict actually what would potentially come out of a genetic cross. So remember, this is the probability of something. It doesn't mean that it will happen that particular way, but given the odds, right, based on what we have for our different types of genes and alleles for those genes, we can predict what we would expect certain offspring to look like. And so you can do this because our alleles segregate randomly, right? We know that during independent assortment that they're going to line up on that metaphase plate in different places, and that because of where they line up, that's going to lead to their final offspring having this mix-up of different genes. So there are some things to note about probability and some assumptions that usually happens, okay, that people think there's like probability, this is how it has to be. So remember that past outcomes in probability have no effect on future events. So this is what's known as the gambler's fallacy. It's this idea that when you're, say, losing a bunch, that if you change one thing, then you'll start winning a bunch. Or if you're winning a bunch, you have to keep doing that same thing because it keeps working. The issue with that is that every single event or every single chance is its own unique event. It's like when you're rolling a die. Okay, so if you roll, say, like 3 threes in a row, you're like, oh, I'm on a streak, so I'm just going to keep going and potentially get another 3. Or you're like, oh, I've had 3 threes, there's no chance that it's going to happen now. But that's not true, because each of those different dice rolls has a 1 in 6 chance of having a 3. And so the next one after you got the 3 threes still has the same probability or the same chance. Okay, so it's just our perception of that. Just remembering that each individual event is its unique own event can help you overcome this idea that because I'm seeing this particular type of offspring, this is going to be what has to happen next. The other thing with probabilities is they predict the average outcome of many events. So they don't necessarily predict what's going to happen with a single event. Okay, we have to look at lots of trials in order to figure out what the probability of that event is going to occur in relation to what we're looking at today. Okay. So when we're calculating probability, probably the easiest way I can represent that is with you flipping a coin. So I don't have any coins with me right now, but if you remember, a coin has a heads and a tails, right? So if you're talking about a penny, you've got Lincoln's head on one and then the Lincoln Monument on the back. And so when you're thinking about the head, there's 2 possible ways that that penny can land. It can either land with the heads facing up or the tails facing up. So that means when you flip it, there are 2 possible outcomes. Again, the head at the top or tails. So when you flip the coin, you have a 1 in 2 chance of getting tails, for example, right? So 1 out of the 2 possibilities, one of those is what you want, one of those is not what you want. So we would say that there's half chance, right? So we can put it as a fraction, we can put it as a ratio, a 1 to 2, right? Or 1 out of 1, because you can get 1 head, 1 tail, right? But when you're flipping the coin, each of those individual events is unique. Okay, so again, if I am going to flip my coin to see if I can get tails 5 times in a row, that means that I would take each of my individual events and multiply them together. So there's a 1 in 2 chance each single time I do it, but every time I do it, I have to add another probability or multiply another probability to it. So in the end, what's going to happen is that for each of my individual coin flips, I still have a 1 in 2 chance of getting tails each individual time. And so the likelihood of that actually happening 5 times in a row, you take each of the individual probabilities, multiply them together, and that will get you your final value. Okay. So thinking about those ratios and the chances of what's going on is useful for us when we're talking about genetic crosses. So in genetic crosses, we use probability to predict the expected outcomes of a cross. right? Based on what alleles an individual has, what's the probability that their final offspring will have a particular genotype or a particular phenotype? We can look at it both ways. So these are used to help us predict and compare what potential variation we might get in our offspring later so that we can understand what traits we might be able to pull out, right? Or the likelihood that we're going to get a particular trait showing up that we might favor or one that we want to get rid of. Okay, for example, we do this a lot when we breed different plant species in order to grow things like fruit, right? We'll start selecting those that have the traits we really like, and we won't select those that have the traits that we don't want. So that say we would want a juicier apple versus one that's really dry. So the diagram that we use to do this is called a Punnett square. Many of you have seen these Punnett squares before, but remember, it's just a diagrammatic view of this probability of our allele showing up. So we use these Punnett squares because it helps us visualize what are the chances if we cross these different alleles together to get our final product. So when we do the cross, the first thing we really have to do is be able to determine what alleles are present in our population or in our individuals that we're meeting together so that we can then figure out what's the probability of those things actually coming together. So remember, each individual, for the purposes of us, we're dealing with diploid things, is going to have 2 alleles for every trait. So when we're talking about our alleles, remember when we had meiosis that we would have 1 copy that came from the mom and 1 copy that came from the dad, and these 2 copies created our homologous chromosomes, right? The ones that coded for the same information, but they might have different versions of that particular gene. So like tyrosinase, we have one that's maybe the normal average one, and then one that's like 2% better at doing its job. So we would say an individual that has 2 identical alleles. So that means that the genes that they got from their mom and the dad are exactly the same, right? That the DNA code is exactly the same. And so we would say that they are homologous, right? And we would say they're homozygous. Okay, so they're homozygous for that allele. They have both of those traits that are exactly the same. Heterozygous are individuals that have different alleles. So remember when we talked about the dominant and recessive, right? They might have an allele for brown, an allele for white, one that they got from their mom, one they got from their dad, and when they come together, you're going to have an individual that has a mix of that. They have both of the genes. They may not both be expressed, but they have both of the copies or types of it. So for an individual to have the expression, right? Remember the alleles are different copies of the same gene. So when we annotate this, we use the same letter to represent that trait. And so you can use generally whatever letter you want. We prefer letters that look different between a capital and a lowercase. So for example, if we're using the trait height, we know that each individual has an allele or 2 alleles for height, one from mom, one from dad. So we would give the dominant allele if they have the one that's expressing the trait we consider dominant from our observations, we give that a capital T. If it's one that's not dominant, it's one that's recessive that only shows up every once in a while, or potentially based on what alleles are present and how they're assorted, it gives us a recessive allele with a lowercase letter. We use the lowercase t in our example for height. So gametes from each of the parents, when we write our Punnett square, and I'll show you what one looks like here in a second, we generally write the alleles at the top and the side, okay? And so we're kind of then filling in the boxes, sort of like if you've ever played like 2048, right? You're just moving the boxes over and you're putting them together. And then we can go through and look at the possible combinations of each of those alleles. So remember phenotypes, when we do a Punnett square, we look for particular phenotypic ratios, like what's the likelihood that you're going to see a particular phenotype. Again, those are the physical characteristics. So if they're tall or short, if they're brown or white. Versus genotype is just where we have our genetic makeup. So what genes does this individual have? What alleles do they have? And what are the allelic combinations for individuals? Are they homozygous dominant? So they have 2 homozygous alleles. Are they heterozygous? So 1 dominant and 1 recessive. Or are they homozygous recessive? So 2 recessive alleles. All right, so we're going to do one. So what you're going to do with a Punnett square is we're going to cross a tall plant with a short plant. So we're going to assume that we have those pure breeding parent plants like Mendel had. And so with the trait we're going to look at and focus on, we're going to just look at height. So our dominant alleles are going to be tall, and our recessive alleles will be those that are short. The parent genotype then, if we have a homozygous dominant parent, is going to be capital T, so 2 big T's. Which is parent 1, so that's the phenotype for tall. And let's say that parent 2 is short and it has 2 recessive alleles. So remember, to be short, you can only have recessive alleles. If you have that T that's dominant, you're always going to express it. So let's do the cross. So we're going to take big T, little t, little t. So here's what that looks like. So I did it really fast. So what you can see here is that I put my alleles at the top and I've split them up in my box. So each individual allele should get its own column or its own row. Okay, and so then when I do my cross, I bring something from the top down, and then I bring this T from the here over. Okay, so bringing this down, bringing this over, and then I can get my final combination. So each box should have a copy of each allele, right? One from each of the parents. So if we look at our ratio here, every individual cell is going to end up with the exact same genotypes, right? They're all going to be big T, little t, and we would say that is a heterozygous offspring. So if we're looking then at the phenotypic expression, each individual cell is going to, or each individual is going to have an expression that is tall, right? And that is because each of them has this dominant allele. Because you have a dominant allele, you will express the trait. So we would say that this particular example is what we call a purebred cross. That's where all of our offspring, because we put two purebreds together, are going to have that heterozygous genotype. So we say we have a 100% chance for it to be heterozygous, that every offspring will have the same genotype and the same phenotype because they have the exact same combinations. So now let's look at a slightly more complicated cross. So this time what we're going to be looking at is what Mendel did in his F1 generation, where he took the plants that he produced from the parents that were all heterozygous, that they all exhibited this tall trait, and we're going to cross those together. So if we think about the gametes for the parents, right, the gametes for the parents are going to be like this. They're all going to be heterozygous. They're going to have a big T allele and a little t allele, one dominant, one recessive. And then for the phenotypes of all the parents, we already know that they're going to be tall because they all exhibit that particular T. So that means our cross is going to be taking this big T, little t, and crossing it with another big T, little t. So for this particular cross, we're saying that we're essentially having a heterozygous cross, right? We're trying to see what the offspring are going to be. So again, we have big T, little t, big T, little t. Okay, and so if we set up our Punnett square like we did before, again with each of the alleles at the top for the parent one, so big T, little t, and then big T, little t. When we look at and bring our things together, so this big T here, this big T here, so that's a big T offspring. Then we have big T and little t, so that's a heterozygous offspring. Here we have a big T and a little t, again heterozygous. And then lastly, we have little t, so homozygous recessive. So then when we look at our results of this particular cross, we have this ratio of probabilities. That when we're looking at the genotypes, a quarter of our offspring are going to be big T, so they're going to be homozygous dominant. Half of our offspring are going to be heterozygous, right? 25% from here and 25% from here. And so they're going to be big T, little t. And then lastly, looking at our genotype for little t, we're going to have 25% of our offspring as that. So if we write that as a ratio, so that means for every one big T we have, we should have 2 heterozygotes and 1 homozygous. So if I have 4 individuals and probability is working perfectly, one of those would be big T genotype, 2 of those would be heterozygous, and 1 of those would be little t. Then if we compare it to the phenotypes, this one, and this one are all going to be individuals that will have that tall phenotype, right? So you'll have an individual who just happens to be homozygous dominant for tall, and 2 that are heterozygous, but they're all going to exhibit that same trait. And then lastly, here's our short individuals. So they're going to be little t, and they're going to be homozygous recessive. So what we've learned from our F2 cross, so again, or the offspring from our F2, is that we're going to see this ratio where a quarter of them are going to be tall with the homozygous dominant allele combination, half of them are going to be heterozygous, and a quarter of them are going to be homozygous short. So Mendel predicted the ratio because he, again, could only see what was happening outside the phenotype that the final seeds were going to distribute. It wasn't until we combined other things about genetics together to really flush out that these alleles were being expressed in this particular manner. So he predicted a ratio of 3 to 1 based on his observations of seeing that when he took this generation about 3 to 1, so like for every 3 plants he had, one of them was short. In that ratio, he just saw this from his observations as he was doing his work. So what we're going to do next is we're going to be looking at Mendelian genetics on a slightly more complicated scale. So we're going to be looking at what would happen, say, if multiple genes are coming together at once, as opposed to what we looked at here, where we're only following one trait. Okay? So if you have questions about Punnett squares, let me know. We're going to be practicing quite a few of these. So if you have questions, shoot them my way.